Hi, this is Devin from Bio 105 Lab, and today I will be talking about the dental disease study that was conducted at UNC Adams Dental School. So, to kind of start off, as far as just background context, a small group of collaborative researchers from two separate universities were a part of this research, specifically from UNC Adams School of Dentistry, primarily, and then also University of Pennsylvania, that conducted an 8 year long project in investigating the microbiome. So, all the microbes like bacteria and how they just play a part in children's mouths that lead to tooth decay. This team was led by Dr. Kimon Devaris at UNC, along with Dr. Hyun Koo as the co-senior author from Penn. And so, for this reason, they tracked over 100 kids at public preschools across North Carolina for many years to collect spit swabs, dental plaque, and water samples to analyze. And so, essentially, the goal for this research was to identify the relationship between oral microbiomes and dental disease in children. So they used certain tools to predict which bacteria broke bad and chomped on teeth. So the few tests they ran were saliva collections, fluoride measurements. Experts also used health statuses of other children's microcompositions to compare and contrast. And lots of testing was done to narrow down the bacteria that contributed to cavity breakdown of the teeth. So after doing so, they collected all these bad bacteria and were able to find and grow them in petri dishes in the lab. And experts were able to compare and contrast between community data to configure the microscopic molecules that crack down tooth surfaces. This discovery was meant to help explain why just killing off the commonly known bacteria isn't the true answer for mitigating tooth decay. And in the end, they were able to discover more unforeseen bacteria that until now played a massive role in pediatric dental damage. And simply with us unveiling these secrets, we're able to build um, a stronger knowledge of dental care and can be more aware of the dangers with bacteria nevertheless. So one thing I thought was interesting from this collaboration as well was specifically Andrea Perel and Jeff Roach contributed to the genome sequencing, sorry, and RNA sequencing analysis, and those parallels aligned with kind of my current teachings in Bio L240. And this correlated into the DNA and RNA transcription and genetic coding of the microorganisms. So as far as the introduction, as a kind of a reiteration, it was simply to find that relationship between oral microbiomes and dental disease in children. Researchers set out to get the full picture of how the community of bacteria in kids' mouths might lead to tooth decay. So they didn't think it was simple as just one bacteria species causing all cavities all the time. Instead, they predicted that lots of different mouth microbes probably work together in some way that make the teeth unhealthy. So more in depth as far as methods. So they took clinical dental examinations, saliva collection used for genetic testing, fluoride measurements, DNA, RNA identification of oral bacteria, microbe confirmation through laboratory culturing, and then also com- researching health status with microbiome composition. Some of the results that came about from the experiment were kids with more cavities had imbalanced bacteria compared to healthy kids. New unexpected germs were discovered, and they tie into the tooth decay in children. A germ called Selenomus butygena was identified as an important decaying bacteria. Groups of bacteria work together to damage teeth as far as just the process of how it works. Mouth microbes increase their sugar munching and acid output in cavities, and then lab tests confirmed that acid oozing trace newly identified tooth decay bacteria. So, as far as just the interpretation, sorry, kids with more cavities had imbalanced uh, mouth bacteria, suggesting dental health relates to the types of microbes present. New germs not typically tied to tooth decay before were discovered to be involved in children's dental disease. And a microbe called, like we've said before, Selenomus butygena was newly associated with tooth damage. Groups of bacteria partnered up in different ways inside mouths to jointly bring on tooth damage. So, as far as conclusions go, they found that certain bacteria groups together collaborate to damage teeth. It's not just about the single bad bacteria causing disease. So, this results in just the acid forming, which dissolves the tooth surfaces and forms gunk, or in other notes, plaque. So this is like a community that leads to tooth damage. And then the imbalance of the microbial ecosystems can give way to illness rather than just one germ being responsible for it based on the microbes children already have in their mouths. And so some future ideas that could be a great addition to the research could be following up with the same group of kids to see how these dental problems in general progress. And this would allow for just ample recordings and trends, maybe patterns to figure out what might predict oral health issues in the future. And maybe also just seeing what other proteins and molecules can cause dental damage just as much as harmful microbes. And so, yeah, that is just the conclusion bit of my dental decay research. Sorry about that, but I appreciate it. Thank you for your time and enjoying this video. And until next time.